Thanks, Lauren. This morning I'm excited to introduce our speaker today, Dr. Brandon Ogbunu. Brandon is an assistant professor of ecology and evolutionary biology at Yale University, where he directs the GEEKS lab, which stands for Genetics, Ecology, Evolution, and Quantitative Science. He completed his PhD at Yale under Dr. Paul Turner, and subsequently did a postdoc at Harvard with Daniel Hartle. Currently, his work looks to understand evolution on both small and large scales. So evolution at the molecular level. So how can we, one of the questions on his website that I pulled out was, how can we leverage evolutionary and ecological theory in design and the use of new drugs, looking at protein-protein interaction networks and constraints on evolution. The other scale he looks at things is on the population level. So how can we disentangle the forces acting on the pathogen, the host, the environment, and the varying interactions between them? And one of his specialties appears to be combining mathematics with empirical data. And so one of his really interesting papers was on looking at empirical lab data to try and predict the emergence of RNA viruses that differ in their host or reservoir use. And then, he really came to my attention really early on in the pandemic through one of our colleagues, Sam Scarpino, where I saw Brandon is really an expert on what I had not heard of before, is the so-called weight SIR models. And so these are models that weight, this stands for waterborne, abiotic, and indirectly transmitted models. And so these are models that basically explicitly model free living survival stage of certain pathogens through an environmental reservoir intermediate. And he's modeled hepatitis C, HCV, cholera, and of course now he's been modeling COVID-19, looking at this, thinking about surface transmission of COVID-19. He also, after that, introduction through Sam, I started following him, and I've really enjoyed following his Twitter account. So that's Big Data Kane, and maybe he will have something on his slides about that, but I highly recommend following him on that. And he is a contributing writer at Wired. And throughout this pandemic, he's had a number of very interesting articles that I would suggest following him on Twitter to come across, looking at the NBA bubble experiment and the success of it in controlling the coronavirus, looking at or thinking about college reopenings, writing about the pandemic or how the pandemic and the protests are kind of mirror images of each other, and then also thinking about coronavirus mutations. And with that, I will, one last thing. I would be remiss if I didn't note that I believe he is our first speaker in our series who has his own IMDb account. And so Brandon was part of a show called Finding Your Roots, The Seedlings, which is a web series that followed 13 young people using science and research skills at a new genetics and genealogy camp to answer the question of, who am I? And I thought that was a nice tidbit, and it looks like an interesting show. So with that, I'll let him take it away.
I mean, that's the talk. As far as I'm concerned, I mean, my goodness, that was magnificent. My mother couldn't have done better than that. That was something else. So thank you, Dr. Fox, for that. That's wonderful. I appreciate that kind introduction. I'm very excited to be here. I'm a little bit Dr. Ansel Miles, I've been a fan of yours for many, many years. So you gave a talk at Yale when I was a graduate student that really changed my career. I remember you were, I was a graduate student then. And you'll meet other students of yours along the way who have been extremely important to my trajectory and who are my closest friends and collaborators today. So I really am excited. Frankly, I think just the thing that I just heard where you went through the literature and then I haven't seen anything like that. That's fantastic. I mean, it's just, wow, very, very impressive. But I know we don't have a lot of time, so let's get through this thing. But I wanted to make sure I properly acknowledged thank you all for the invitation. And I'm excited to, I'm just going to drop a few kind of provocative thoughts and leave. That's the thing about this format. I can just say a few things, and then that's the great thing about Zoom. I can leave. So that's what we're going to do today. The title is Identifiability and Indirect Transmission, Positive Lessons from a Probably Negative Result. This covers kind of two pieces of papers, as many of you know. The companion piece is actually out. The original piece is not yet. You know the way that works. So these are two papers that we worked on that are involved in environmental transmission. But some quick primer points. Indirect or environmental transmission remains a relatively underappreciated aspect of the ecology of infectious disease. I think I'll get no fights there. COVID-19 presents some unique challenges with regards to understanding this problem. Negative results can be useful and fruitful, but we need to be careful with how they are sold. I think that's one of the things. So I think about my introduction to the ecology of infectious disease. This is me at 17 years old. And that facial expression is for two reasons. I'm proud because we shut them out 24-0. And I had a big game, a bunch of tackles and a sack and a half, I think, or whatever. Maybe I'm inflating my stats a little bit, but I remember I was happy. But the point is, I'm not all happy because all of the linebackers at this point are suffering from a bad case of tinea pedis. We have a bad outbreak in the locker room, including this, and of course we did what any responsible and thoughtful 17-year-old would do at the time. I wasn't much of a student in high school, so I blame the hockey team. But of course, it was much more complicated than that. And so we went and we actually addressed this as something with the team. And I turned back. This isn't actually my coach. This is Denzel Washington from Remember the Titans. But I had a defensive coach who was a lot like this person. And frankly, who I used to worship. His name was Coach Al. And what he said was, watch out for the shower. There are nasty things on the floor waiting for you. And he actually kind of gave us like lectureship on hygiene and was really my first ecology of infectious diseases teacher. Ended up kind of saving our season. We finished 4-4, but whatever. But the point was, I actually, as a homage to Coach Al, many years later, developed this theoretical paradigm called waterborne, abiotic, and indirectly transmitted infections. Now, I'm not the first person to do this. I'm not the best person to do this. This has been done for many decades. Many smart people have worked on better versions of this. Frankly, I developed this as a pedagogical device for these two former physicists, undergraduates who were in my lab to teach them, right, how to model. But nonetheless, that just became something that we worked on. And my interest in kind of indirect transmission and free living survival actually goes back to the Turner lab. And, I could talk about that at another time when I was thinking about virus evolution. But nonetheless, this became kind of a framework that we use to think about infectious disease. Of course, right, my interest in a lot of these diseases, the things that I think about in particular influence are the times they were in. The same way young people now are going to be interested in COVID forever. I'm like a lot of exennials, right? I think the Magic Johnson announcement was a very defining point for me. And of course, I'm also from New York City, and I grew up largely during the crack epidemics. I remember the influence of the injection drug use epidemic, that one from the 80s and 90s. I remember that role that played in my life. So these things have all influenced my choice of problem. But of course, now there's even more recent ones. And so I think you think about kind of how HIV/AIDS framed my interest, and you think about the way that the problems that kind of caused when it came to injection drug use. So injection drug use and these kind of things have always been kind of important kind of problems for me and questions for me. And we actually took the weight model, right, and we applied it to something, right? We applied it to hepatitis C virus. This is an old slide. Of course, everybody knows it now because it just won the Nobel Prize. but I had been thinking about this for some time. And hepatitis C virus, you don't have time to get into it right now, but it's a big problem. It's called the silent epidemic, both because of the way that of the natural history of the disease, and because of the population who it affects. It's a population people don't care very much about. So when you put those two things together, we call it the silent killer for that reason. So we took our weight model and we applied it to understand kind of the way an epidemic, spread. And basically what we do is we examine models of injection drug use, and we found a fitting kind of analogy in the mathematical modeling literature. And that comes from the vector literature, right? I mean, there's obviously differences, but there's some similarities in ways that we can kind of anchor the understanding of injection drug use to vector-borne disease. And that's exactly what we did. This was published last year in 2019. Basically took that weight framework, which was just a preprint that kind of fleshed out the basic concept of environmental transmission, and we basically applied it to the hepatitis C virus, what we knew about hepatitis C virus natural history, And we kind of revealed some specific things that, like, why is it, or how much would safe injection be more effective than the needle exchange, right? We can kind of introduce these things about harm reduction, things about therapy, I think, which is built into this model. So all these kind of different features of it. People have looked at this before, largely in HIV, but not for some time ago. It's been a while since this was done rigorously for this type of problem. And we did it for hepatitis C virus. So that's just kind of giving you an example of the kind of ways that we've adapted this model. Originally, that started on the floor of my locker room, my high school locker room shower. And I've kind of adapted it, right, my inspiration from Coach Allen. I've adapted to kind of think about this. I've also done this for cholera. So I do this also for waterborne disease, which also kind of has a lot of these features. But then, of course, right, this happened. And I said, all right, well, I think when that initially happened, I think we all felt emboldened, and none more than this group. And, few have done better group work on this than you all. But it's like, what do we have to say about it? Which the thing that, and I kind of was, I was kind of reluctant to jump in until I had something smart, I thought, or to add to the conversation. So the first set of things that I did were really in the public engagement space. Some of you, frankly, I think like these videos we did on what is the R naught and stuff like that with Professor Paluni Pennings. I don't know if you know Paluni. Paluni's magnificent, one of my favorite people in science, and Sanaya Barg, now at UNC Chapel Hill. We did this series just kind of explaining some of these basic comments. We've actually had to update them quite a bit because some of our stuff wasn't quite right early, but but these ended up getting millions of views. This is probably like because I think we kind of were early on, some of the scientists that were translating some of these basic concepts. But some of the things that we were translating, right, were things about like, how is this actually transmitted? And most of the stuff's actually stood up reasonably well. So it can spread through casual contact. This is Paluni did this. I don't know how she does this on her iPad Pro. When I draw it, doesn't work quite this way. In close proximity, and this is from like April, and this is mostly stood up, right, to tell, sneezing and coughing and creating droplets. Of course, it's also talking loud, right? There's some dimensions that I've added here, but this is kind of mostly stood up to scrutiny. But then, of course, right, there's some other details. This came out, of course, right, and I think this is kind of one of the loudest papers from the SARS-CoV-2 era that came out in April, and it talked about the surface stability of SARS-CoV-1 and SARS-CoV-2. Now, of course, it compared SARS-CoV-1 and CoV-2, but the real comparison, of course, was the survival of the different SARS-CoV-2 strains on these different physical surfaces, or in the case of aerosols, in this different physical media. And of course, this led to a lot of kind of hysteria. Remember when we were all nuking our Amazon packages and all that, and pizza, and we couldn't, right? And when it came to pizza, I didn't care. I was like, I just got to get this. I got to have my pizza. So, but the point was, it led to a lot, but fair in some ways, because this paper basically said that this thing can travel on surfaces and it can survive on different surfaces for different amounts of time. So when I saw that, I said, all right, well, fine. Now it's time for me to say something, because I just told you, going back from Coach Al all the way to hepatitis C virus and cholera, I think carefully about the way viruses are spread on surfaces. So I said, fine. So I talked to, right, y'all boy, okay, right, one of my closest friends and colleagues, and I love Sam for many reasons, but I think Sam is kind of like he's brazen in this way that I wouldn't be, and I think that's really important for young scientists. Like get a colleague who's brazen, who kind of who just says things. I'm very reluctant, and imposter syndrome and stuff. Sam definitely does not have that, right? In addition to being very brilliant. So the point is, I think that's good. So we talked to Sam, and I brought my crew in, and we said, let's get on this. Let's think about a careful and something we can say about surface transmission. So what do we do? You look at the Hcv model, right? We basically almost did the exact same thing. Now there's some particulars that are slightly different, because we don't have here. We have this kind of dichotomy between infected and uninfected needles, and that relates to the instrument of infection. We wanted to we were very careful. We wanted to know very specifically what happens when you swap uninfected for infected. So due to the question, right, we engineered this. But in both these cases, you have this environmental reservoir. Okay, and I think this model, and we'll get into kind of this was kind of at the time was the one that best fit, and I think probably still does. The natural history of disease, where in terms of the compartmental structure, and we used the model that we're kind of coming in about, the rates, the incubation period, and we use all that. We can get into this, but we used all that to kind of bake in this model, this weight kind of iteration of COVID-19. And there were very few, if any, at the time that did that. So, right, we looked at this and we basically used this as a guide. We said, okay, well, if I told you here I have this reservoir, and we said, all right, we have this data for the way that SARS-CoV-2 was surviving on different reservoirs, right? What we did was we just took the average. Okay, well, let's just say the world is kind of composed of some kind of average of these things. Let's just give that as an average surface that people are interacting with. And we use that to build this version of the model. And we basically fit this model. We fit a model with the weight compartment, with a model without the weight compartment, and we fit it to a bunch of different kind of early country outbreaks, right? And and these are actually, so this is Spain. These are the 4 of the biggest early epidemics, actually, but we have this for 17 countries. We have it actually for much more now, but we use for 17 countries. And what you see is the IIC, as you all know, is a measure of the fit. So basically fit better. The weight compartment model fit the outbreak data better, than the non-weight variant. Now, I know that doesn't mean anything. We're gonna get there, I promise, right? Or rather, it doesn't necessarily mean that's the way this is working. And I think there's an art to kind of fitting model data rather, that you have to consider here. But what that did tell us is, okay, we're not crazy for asking this question. It was kind of like some support that, okay, let's continue to dig on this environmental and indirect transmission phenomenon. So, we just did a lot of this stuff. I think this crowd understands this, but I just kind of showed what some of the things that we did. We basically peeled apart the R naught into these 2 different components. You can see how the different parameters are. This is direct infection. This is environmental transmission. We can actually do sensitivity analyses across the different subcomponents. Environmental transmission can be defined in a separate expression. This is stuff that, certainly Professor Anson Myers helped come up with a lot of the stuff. So I don't need to tell this group, but the point is we did the basic kind of stuff you're supposed to do. But then when I thought, okay, well, now that we've done the basic set of things for this model, it seems to fit data in some way. What's a bigger theoretical question we can ask about surface transmission with regards to SARS-CoV-2 that might kind of lead to more provocative, and it may or may not directly apply. I was very reluctant to kind of say anything particular about this disease, but it's like, how do you walk that line between something theoretical but without being too brazen, right? That's the thing, right? But you want to say, you think you have something to add, but you don't want to say anything about what people should be doing with the Amazon packages necessarily. And I think that's a hard line to walk in this pandemic, because even when you have honest intentions, they can get misconstrued. This is a huge point of, I don't know about anxiety, but discussion in my group and Sam Scarpino. So what it is, like a lot of us, I think I was binging on TV during quarantine, and sci-fi fans. So I saw some heads nod. Good. Okay. So when I say, what's the largest movie flop of all time? What movie comes to mind? Usually people get this. Some people, when I say flop, like I say the movie all-time flop, and I think some of you are a little young, but if you're anywhere near my age, which you would understand this. But basically, you're going to see it. You're going to be like, of course, it's going to be Waterworld. Okay. It's gonna be Waterworld. That was an all-time flop, box office flop. Now, this movie was a disaster. It cost like $1 gazillion and didn't make very much. And I watched this movie and it was bad, not quite as bad as I remember, frankly. I think, and the premise of it was quite interesting. But when I watched this film, in the midst of all this, I got an idea for a theoretical question we can ask in the context of weight transmission and SARS-CoV-2. I said, instead of a water world, what if we modeled this disease as if it was being transmitted, in a set of worlds that look like this, but were all copper, or were all made of steel, or were all made of plastic, or were all made of cardboard. The idea here is the surfaces that the world is composed of is all that thing. It's a theoretical question. And if you have surface transmission, what do the disease dynamics look like in a large population, right? And I think that kind of tells you, it's a way to kind of push this thing to its theoretical bound limits and ask some basic questions. Now, I'm not going to ask you to disentangle this. I think the more useful figure is this one, right? Is this one, okay? Right, we basically look at some basic features of the epidemic, and we say some things like, all right, well, what does this actually look like? I mean, look at that or not. Those are 2 different diseases, right? In this realm, right? So in a world where all the surfaces that we interact with cups, doorknobs, gym mats, everything that we kind of interact with as people, if it's all plastic, if this disease, this is a key question here, if this disease can truly be transmitted via surfaces, key caveat, okay? Key caveat. If this thing can truly be transmitted via surfaces, and the parameterization for this comes from a lot of other virus transmission, by the way, which is another caveat. Right, a lot of the stuff is a point of controversy, which we'll talk about. But if that's the case, I mean, the R0 here is 3.385. Look how much lower it is. Also, no, I think interestingly enough, look how close the dynamics are between aerosol. Now, what does aerosol mean? Aerosol simply means that there's effectively no environmental transmission. Now, the only indirect transmission there is from individuals being able to interact with air that other individuals have interacted with. It's not like a physical surface. 30-day total is just the number of cases, right? We see that. Imax is the number of the peak number of infected individuals, whatever that peak is in the first 30 days. And then Tmax is how fast it reaches that peak. So it's not just that on plastic. I mean, a lot of this is kind of embedded in the R0, of course, but the point is it's kind of, it's intriguing to pull this apart and be able to look at this. So the idea is you get these really, really different, if in fact this is happening, you get these different dynamics. All right. So, of course, the skepticism that's arisen around this, and I think this has been a point of debate the entire time, right? I think this, the way I discussed this with the research group, the role of foam-like transmission, it just comes and goes. I think the overall sentiment, right, is that it's not the main route of transmission, right, or it's not a major route of transmission most places. In fact, I said as much, right? Right. So you see here, right? The transmission through abandonment services is very, very small. I think that's kind of where we've landed on the project on this, and we know that, right, the environmental transmission model fits outbreak data better than SEIR. We can identify terms which specifically influence environmental transmission, and I think like I said, I'm fine with it being a negative result. That's probably a little extreme because we don't think we have necessarily argued any one thing, but fine. I'm fine with the notion that that's probably not the case in terms of its being a main route of transmission. But, right, again, these two things keep popping up, right? Remember the New Zealand outbreak down in Shirley you talked about in this group, right, where they actually think, I don't know if I haven't followed up with this, so I don't actually know what the conclusion was, but the speculation there was that I actually might have gotten in via freight. Of course, I actually, I actually went against my own work and I said, probably not. But we don't know here. It was quoted in this article here. So I don't make any connection between this and any fear. So I think my formal stance has been, when I've talked about this formally, that no, deep cleaning is not a good idea. No, we don't need to microwave and nuke everything that comes in, and no, you shouldn't fear Amazon packages, right? But nonetheless, these things keep popping up. This is last week, right? And you probably talked about this. So basically, it's a lot of news, and of course, the news took this and went too far with it, I think. But again, now there's new evidence that maybe infectious virus can that surfaces can carry infectious SARS-CoV-2. I think more than that, however, is kind of what I think, and the way we've gone with this work, me, Sam, and colleagues, is thinking about the challenges of very, very particularly resolving a route of transmission when you try to fit models to data. And this kind of relates to something that, Joshua Weitz and others have worked on called the identifiability problem. And what they basically said is that basically what it comes down to is for any data, you can fit a lot of models to a data, and a lot of them can fit them kind of with relative accuracy. And so it's really, really hard to say for certain whether the route of transmission is one way or another based solely on epidemiological time series. And I think, frankly, this works just as much for environmental transmission as against it, theoretically, right? But it is kind of one of the things where we kind of have to be very, very sure and careful, right? So we think about these fits, right? You think about the fact that the weight models are fitting better, and I think they fit 10 of 17 countries better. And now we have 90 countries. And we're actually going to, we're writing a short, me and Sam are going to write a short perspective piece on writing a short perspective piece on identifiability, where we have a larger swath of countries. Yeah, I mean, the weight model fits most of them better. Okay, most of them better. But here's the key, and I think we thought carefully about this. Maybe the lesson isn't about environmental indirect transmission. Maybe this component is just kind of a composite of all of the funky dynamics that we don't quite that kind of describe SARS-CoV-2 transmission in different settings. Maybe that's why it's fitting. Maybe it's not anything particular about surfaces. Maybe they're kind of as we've observed, from super spreading to all these other contextual things that are kind of explain the variance in the way this thing is spreading in certain settings. Maybe kind of this kind of by happenstance captures some of those dynamics. So nonetheless, I think this is it's been a useful thing for us to think about, because it is something that we're continuing to think about with regards to SARS-CoV-2 and of course other infectious diseases.